Hi everyone, my group consists of me, Daniel, Hani, Shami, and Yushaiman will present molecular techniques in conserving biodiversity. In this video, we will be discussing cloning, in vitro propagation, genetic monitoring, and other key techniques that protect species and preserve diversity. Let's get started.
Hi, I'm Badran. I'm going to talk about molecular monitoring and genetic diversity assessment, which are vital techniques in conserving biodiversity, providing detailed insights into genetic health of species. By analyzing variation in DNA, we can identify populations at risk and implement targeted conservation strategies. For example, studies on coral species have shown that genetic diversity is critical for their resilience to warming seas, while similar research on bird population has helped manage responses to emerging diseases. A global study involving 919 species revealed that over half face decline so severe they cannot maintain their genetic diversity, underscoring the urgent need for action. Techniques such as DNA barcoding allow for precise identification of species and their genetic structures. Research on the giant panda exemplifies this, as genetic markers were used to design habitat corridors that enhance genetic diversity in fragmented populations. Similarly, wild rice genetic monitoring has preserved traits essential for crop resilience and habitat restoration, showcasing the practical benefits of these tools. These examples highlight how molecular approaches strengthen conservation efforts for both animals and plants. Globally, the coming Montreal Global Biodiversity Framework has prioritized conserving genetic diversity, with 196 nations adopting strategies based on genetic health indicators. This ensures more inclusive and effective conservation, even in regions with limited resources. Genetic diversity is more than a metric. It is the foundation of ecosystem adaptability and resilience. By advancing molecular monitoring, we safeguard the biodiversity and ensure species survival in an ever-changing environment. Thank you.
Hi, I'm Daniel, and let's move into the next method that named cryopreservation. So, what is cryopreservation? It is a method that used to freeze and store living cells, seeds, or tissues at very low temperature. So, it's usually being applied in liquid nitrogen at about negative 196 degrees of Celsius. So, at this extremely low temperature, all activity in the cell are being stopped by keeping them alive and also being usable for a long period. And there are two main ways actually where this can be done, which is by the process called slow freezing and also vitrification. Slow freezing is much simpler than vitrification as it just can be applied directly to for preserving the sperm. While vitrification is a bit complex and very suitable for embryos and eggs because it can help to prevent the formation of harmful ice crystals. So this method, that name cryopreservation, is especially important for some species that are hard to be saved from extinction by just applying regular methods of storage or any other kind of in situ approaches. For example, is a wild banana that grows in Southeast Asia, where their seeds are being cryopreserved in order to protect their genetic traits, which are very crucial in order to develop stronger and also disease resistant banana crops in the future. So for animals, there is some species that being applied to this method, uh, like European eel, where scientists have frozen their sperm, which could help to save the genetic material from endangered situation. So the European eel, which is endangered because of the overfishing activities and also habitat loss. So this cryopreservation of their sperm could help a lot in breeding programs to save their species. And also for your information, cryopreservation is actually the backbone of genetic banks. We have all heard about genetic banks, but yeah, cryopreservation help a lot by protect the genetic material to ensure the future survival of species and provide a backup plan if there's some species that going to extinct in the wild. And it also help the breeding programs by increasing the genetic variety and reducing the inbreeding cases, and also help to support the research for better understanding regarding some species, and also and even help to restore them for the future usage. So that's all from me. Let's continue to the next method.
Hi, I am Hani and I am going to talk about cloning for conservation. So what is cloning? Cloning is the process of creating genetically identical organisms through somatic cell nuclear transfer SCNT. So the goal for cloning is to preserve genetic diversity, revive endangered species, and support population with low genetic variability. So one of the most famous example of cloning is the successful cloning of endangered species like bucardo and black-footed ferret. So, how does cloning works in conservation? It is by using somatic cell nuclear transfer (SCNT). So, firstly, we need to do the donor nucleus extraction, where a somatic cell from the endangered species is extracted. Then, an egg cell from a closely related species is enucleated, where we remove the nucleus of the cell. Then, the donor nucleus is fused with the enucleated egg cells. Next, the embryo is stimulated to divide and develop in vitro. And lastly, embryo is implanted into a surrogate mother for gestation. Now let's move to the advantage of cloning for conservation. Firstly, it can preserve genetic material where cloning can help maintain the genetic diversity of critical endangered species. Next, it can rescue the extinct species where it offers the potential to bring back recently extinct species using preserved genetic materials. And lastly, it is an existence in captive breeding, where cloning supplements traditional breeding efforts in zoos and research facilities. There are few challenges and ethical considerations while using this method. Firstly, it is a low success rate, where many cloning attempts fail at various stages from embryo development to birth. Next, genetic bottleneck, where clones do not add to genetic diversity, thus over-reliance may lead to reduced variability. Lastly, ethical concerns where the welfare of clones and surrogates and long-term ecosystem impacts. As for conclusion, cloning is a promising tool for biodiversity conservation, especially for species on the brink of extinction, and it should be used in tandem with other conservation methods like habitat protections and genetic banking. That's all from me. Thank you.
In vitro propagation refers to the cultivation of plant cells, tissue, or organ in a controlled, sterile environment on a nutrient-rich culture medium. The rules in conserving endangered plant species is to generate a large number of plants from a single explant in a short time. Next is to sterile and control condition, which is to ensure the propagation of healthy plants free from pathogens. Then is conservation of genetic material like germplasm that can be preserved for long-term storage and propagation. Plants can be propagated irrespective of seasons or climatic conditions. Lastly, propagated plants can be reintroduced into their natural habitat. For example of plants that include in this in vitro propagation is orchid, which are one of the most threatened plant groups due to their slow growth and specialized habitat requirement. Next is medicinal plants like Rauwolfia serpentina, which is valued for its alkaloids and endangered due to overharvesting. These are the books that I used as sources, and I recommended it to discuss more about conservation of endangered medicinal plants and preservation of rare plants, which is using tissue culture.
Hi, my name is Simon Hakim. Now we move to DNA barcoding. Application in identifying and catalog species. First one is species identification. DNA barcoding helps differentiate species that are morphologically similar or cryptic, those that appear identical but are genetically distant. For example, is frosted tiger beetles. Second one is cataloging biodiversity. It is a critical tool for documenting life on Earth, aiding in the discovery of new species and updating existing taxonomy classification. The third one is monitoring ecosystem. Barcoding provides insight into ecosystem health by analyzing environmental DNA or eDNA from soil, water, or air samples to detect the presence of various organisms. Next, we move to the role in combating illegal wildlife trades. The illegal wildlife trades are major threats to global biodiversity. The first one is authenticating species, identifying protected endangered species in seized wildlife products or derivatives in ivory, leather, or traditional medicine. Next is exposing supply chain, tracing the origin of the traffic, animal and plant. The third one is improving regulatory measures, empowering custom and law enforcement with the reliable, fast and cost-effective identification tools. The last one is protecting unknown or cryptic species. DNA barcoding is vital for discovering cryptic species that are at risk of extinction due to habitat destruction or overexploitation. It's enabled targeted conservation strategies by revealing previously unnoticed biodiversity. For the mislabeled fish in market, a notable application of DNA barcoding in detecting mislabeled fish species in the market and restaurant in a study conducted by Oceana, DNA barcoding was used to examine fish sold in the US market, revealing that approximately 1/3 of the sample were mislabeled. For example, the first one is red snapper, a highly sought after fish was often replaced with cheaper, less sustainable species like tilapia and rockfish. Next one is health implication. Mislabel can inadvertently expose consumer to allergies or harmful toxin present in the substitute species. Lastly is the conservation impact. It exhibit overfishing of vulnerable population while under aiming consumer trust and sustainable seafood practices.